Thanks to our colleagues from ARM for inviting us, and good morning to everyone. I look forward to sharing the story of Vesigen, which is a story of unlocking novel therapeutics with innovative non-viral delivery. As everyone in this room probably is well aware that we have all kinds of clever ways to edit biology in the biopharma R&D community, whether it's gene editors, base editors, prime editors, RNA therapeutics, protein effectors and degraders. The list is long and goes on and seems to be growing every day. But all of these methods struggle when it comes to delivery. Right? Whether it's the redosability challenge, the cassette size limitation, these are the things that plague AAV. Whether it's, in the case of LNPs, getting to the liver, maybe a thing that can be done. It's a trick otherwise. There's been reports of toxicity as well. CMC is a challenge for all of these. These are the problems of delivery, and they're well known and they have not yet been solved. Our vision at Vesigen is to get past these problems and allow some complex therapeutics to be delivered very efficiently to very specific cell types within very specific organ systems. And let me tell you how we do that. We use our human ARMS technology, and I'll explain that acronym on the next slide. But let me point to several key attributes of ARMS. First, it's redosable. It's fully human-derived. There's no neutralizing antibody generation, and imminently redosable, and we have lots of experiments proving that. The cargo capacity is very large, so not only can you fit Cas9 plus a guide, you can fit some of the bigger base editors and newer modalities as well. In terms of tissue targeting, you can get to virtually any cell type out there using either the natural biodistribution, which is unique, or adding surface decoration to get these particles to your chosen target of interest. The delivery is very efficient compared to other vehicles. The loading is very efficient. I'll explain that a little later. And on the recipient cell side, the vehicle does not get sequestered into an endosome and suffer the endosomal trap phenomenon that other vehicles suffer. And lastly, the CMC process is quite straightforward, and that's not something you hear typically when it comes to some of these therapeutics. If that list sounds too good to be true, bear with me over the next several slides, and I'll give you some reasons to believe. So let me start with what are ARMS, and how is it that we're leveraging this national biology. So ARMS it stands for ARDC1 mediated microvesicles. That's a mouthful. It refers to a class of human-derived extracellular vesicles, which were discovered at Harvard. The A in ARMS stands for ARDC1, which is a naturally occurring protein, and its responsibility is biogenesis of this class of vesicles. So you see in the picture there, ARDC1 is depicted by these little green triangles, and ARMS are the vesicles, which you can see budding from the cell surface. Their native function is intracellular signaling and transport, and importantly, there is no viral component or viral heritage. These are completely immune silent. What we do at Vesigen is we co-opt or hijack this ARMS process for delivery of therapeutics. And the way we do that is laid out on this slide. I'll start at the beginning. Step number one is we take the sequence for the payload of interest, and we append that to ARDC1. So you see ARDC1 in green there, and the payload in blue. They're linked together in orange with an engineered tether. So when the cell manufactures ARDC1, it does so conjugated to the payload, which is depicted by that blue oval. And the payload could be a gene editor, it could be a protein, or it could just be the RNA. Step two then, when the producer cell line is creating all these ARDC1 proteins, you see them tethered there to the blue, and as the vehicles bud, they drag the payload into the intraluminal space of the vesicle. So it's sort of safely nestled in there, protected by this natural lipid bilayer. And then what you do is you'd isolate these by size, and that becomes your therapeutic. And you see over to the right, the ultimate goal would be to administer those to humans. We're getting pretty close to that with some very recent readouts that we're excited about, showing that we can do this process quite effectively in non-human primates. I'll show you a couple examples, both in the eye as well as systemically administered. Well, let's talk about, for a second, how to get to the tissue type of choice. As I mentioned in brief, there's two ways to do that. One is you can take advantage of the natural biodistribution and really just vary the route of administration. So you can do something systemic, you can do intraocular, intranasal, intrathecal, et cetera. We've spent a lot of time mapping exactly where these particles go, and we have a nice understanding of that. Or if the cell type is not on that list of natural distribution, you can add surface decoration. So by adding some targeting moieties to the surface, you're able to access very otherwise hard to reach cell types, T cells, B cells, HSCs, et cetera. I'll give you three examples of this, two in the first category of natural biodistribution and one in the second category of retargeting. I'll start with the eye, and in the eye, if you do an intraocular administration, these particles get taken up by two cell types. That's photoreceptors and RPE, or retinal pigmented epithelium. You see on the left the different species. This data translates nicely from mice to an intermediate species, mini pigs, and ultimately NHPs. The data here is from NHPs. The top part shows some microscopy from the cones, which is one of the types of photoreceptors. The top row there is a control, and the second row is arms loaded with a red reporter called M-cherry. So the first column stains for cones, so you see those are going to be the same. The second column stains for the reporter, so obviously there's none in the control, and in the second you'll see the exact same places line up as you see in the first column. So the third column is a merge, and if you look at that inset in particular, you'll see some very clear co-localization of the M-cherry signal. That's the top, and in the bottom half, you see a cross-section of the entire globe of the eye. And if you can make out with the colors there, you can see where the arms light up in the back. And this is a very smooth and even distribution. There's no toxicity in hotspots. If you were to do the same experiment with LNPs, you would see sort of a splotchy distribution there with sort of bumps that light up. We've also quantified this biodistribution. 80% of the photoreceptors take up arms, and 100% of RPE cells take up arms, so pretty robust biodistribution. So this chart, of course, is biodistribution, i.e. can we get to the right cell types? We've also done a separate NHP study showing that we can not just get there, but we can actually deliver a base editor there. And this translates nicely from smaller species like rodents all the way through NHPs. So that's pretty exciting, right? So we can get to the right place, we can do the edit. That gives us pretty good confidence that we can tackle some of these inherited retinal diseases. So for example, Stargardt's disease, AMD, geographic atrophy, there's a lot of diseases, inherited and otherwise, that you can tackle once you have sort of safe access to this particular compartment. Switching gears to systemic delivery, so this is an IV introduction of arms. Again, I'll note that we have done several experiments, both studying biodistribution as well as studying gene editors. You get to two cell types in the liver. So liver is a typical destination, although we don't get to hepatocytes. So hepatocytes are the cell population that are taken up by LNPs, by exosomes. We don't see any uptake in hepatocytes. Instead, we see uptake in two other cell types. That's Kupffer cells, so the liver-resident macrophage population, and liver sinusoidal endothelial cells, or LSCCs. So on the panel on the left, you see some biodistribution, again, some microscopy, and you see kind of in green are the LSCCs, in red is the M-cherry, so you see very kind of clear co-localization. Again, this biodistribution is translated very nicely across species. No observed toxicity. In fact, we've not seen toxicity in any experiment we've run for the history of the company. We've also done a gene editing study, which is not pictured here, but we can effectively edit LSCCs. And the reason that's important is LSCCs are the only cell type that expresses factor VIII, right? So this starts to suggest some pretty interesting options for hemophilia, driving to a cure, as opposed to regular introductions of enzyme or gene therapies, which may have durability limits. On the right panel, this is a gene editing couple of charts. Here we took arms in this experiment and we loaded it with Cas9 plus a guide targeting NLRP3 knockout. NLRP3, for those that may not know, is a well-validated node implicated in a lot of immune modulation conditions. So you see that 60% gene editing in the Kupffer cell populations. So again, macrophages tend to gobble lots of things up, but here you're not just gobbling up, you're actually editing the macrophage. And 60% is, an order of magnitude more than anything we've seen published. Pretty excited with those results, so we tried this in a disease model. It's a mouse model for acute liver injury. So on the bottom chart, you see the control mouse on the left. So you introduce a liver injury in the form of carbon tetrachloride. You see a huge spike in liver enzymes. ALT is pictured here. We have similar charts for AST and others. And the mouse that was injected with ARMS with this NLRP3 knockout, you see a dramatic blunting of the liver enzyme spike. So this opens up a lot of interesting possibilities when it comes to immune modulation. I'm going fast here just in the interest of time. One of the topics that has garnered a fair amount of enthusiasm from a lot of the folks that I've spoken to is this notion of being able to target these particles to very specific cell types of interest. We started with CD8 positive T cells as a target because they're notoriously difficult to transfect. So we did a couple experiments here. The one on the left, you see our ARMS particles with the red inside. That red is the fluorescent reporter M cherry. And you see these little orange decorations on the surface. That targeting moiety in this case is a single chain variable fragment. And what we did is we introduced this to a mix of immune cells. And you see in the chart on the left panel, the non-targeted arms show virtually no uptake, and the targeted arms show dramatic uptake. So again, pretty excited with this and the fact that we can now conceivably get to T cells. Now we wanted to make sure we're not just getting there with a red reporter, but can we actually deliver an editor? So we did a very analogous experiment on the right, same arms and same targeting, but inside them this time was and you see with this chart a very similar result. So CD8 positive cells get huge uptake. Other cells like CD8 negative, you see virtually none. And look, it's always exciting to see stuff in a dish, but it's nothing like seeing it in vivo. This is a brand new data for us where we are able to show that in a mouse, again, same experiment, non-targeting versus targeting, you see both in the blood and the spleen, very clear results showing we're able to see uptake in the CD8 targeted ones, and in the non-targeted ones, we see no uptake. So this shows us we're able to robustly target T cells using ARMS, and we don't see uptake in other T cells. We've done this now with lots of different cell types, CD8, CD4, HSCs, B cells, some flavors of neurons, fibroblasts. We're pretty excited about this, but we're at the level where we'd love to consider partnering. So if other entities are out there that have particular cells that they'd love to get to, we're pretty sure our system is robust enough. It's really just a question of, can you find the right surface decoration, the right engager to. A word on manufacturing. This is actually a fairly straightforward process. It has all of one upstream step, and that's really taking the sequence for your payload of interest and knocking it into a safe harbor locus. That is it. The cell does the rest of the work. So the cell is actually doing all the loading for you, which is that efficient loading that I mentioned at the beginning. Contrast this with other platforms. You have a much more complicated upstream step. You have to make the vehicle, isolate it. You have a separate scheme to make the payload, isolate a separate stream yet to actually conjugate them together. Whereas here, kind of one step and done. And the downstream is fairly straightforward as well. Separate by size, separate by charge, polish, fill and finish. So pretty standard unit operations, starting with off-the-shelf materials, makes for a process which is A, scalable, and B, easy to contemplate with a third-party solution. So where does this all put us? Our emerging pipeline looks something like this. We prioritize the eye because we have the NHP data in hand, both by distribution and gene editing. A disease like Stargardt's makes a lot of sense. It is genetically very well defined, doesn't carry that much biology risk, but it's a reasonably large population. I mentioned in the liver there's some interesting options, and again, with this notion of ability to target tropism, we can really get to a lot of different places. Last slide, I'd be remiss if I didn't mention, I'm here only because the team is pretty fantastic and has been able to accomplish a lot, also with the support of our investors from Morningside and Bayer. That, it looks like I'm just that time. So thank you very much. Appreciate your time.